Good day, ma'am. We are the Group 10 and we are assigned to report the recombinant DNA technology. Our topic comprises the definition, the example of recombinant DNA technology, the steps. It also includes the discovery, the limitation, and the applications of the recombinant DNA technology. To start with, let me introduce to you the definition of recombinant DNA technology. So, recombinant DNA technology is joining together of DNA molecules from two different species. So we know already the molecules that it is the two or more atoms together to form a single chemical entity. So the recombinant DNA technology, this is the molecules that join together from two different species. The recombined DNA molecules is inserted into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. So, the genetic combinations, it is the genetic methods in it that exchange to the different chromosomes or between different regions within the same chromosome. So, the recombinant DNA technology in the field of science, scientists make a research, or they make a study so that they can produce two new genetic informations ilang recombine para maka-produce sila og genetic na pod nga bag-o na pod or new genetic recombinations. And then in the field of medicines, just like for example the vaccine. So as of today, we are acquired, or especially to us as students, we are acquired to get vaccinated so that we can kanang maka-avail na ta sa new normal class nga which is face to face, so that the vaccine kanang i-inject sa tua para makuha na ta, we are free. So let's proceed the example of recombinant DNA technology. So the example that I put here is insulin. So insulin, a hormone made in your pancreas, a gland located behind your stomach, and it also allows your body to use glucose for energy. So that is the definitions of the insulin, that it is located located behind your stomach, and it is allows the body to use glucose so that we can gain energy. So how did they make insulin from recombinant DNA? So these following steps, the 7 steps, teach us on how to make insulin from recombinant DNA technology. So first is scientific build the human insulin gene in the laboratory. So they remove a loop of bacterial DNA known as plasmid. Then second, insert the human insulin gene into the plasmid. Then third, the researchers return the plasmid to the bacterial. And for the fourth, put the recombinant bacteria in large fermentation tanks. And the fifth steps, the recombinant bacteria use the gene to begin producing human insulin. Sixth, scientists harvest the insulin from the bacteria, and lastly, purify the substance for use as a medicine for people. So these following steps can produce a new product, which is the medicine for people. This is the product that the scientists make insulin from recombinant DNA is the medicine for people. For example, the paracetamol medicine, or we have a lots of medicine. So that medicines, that medicines made by the scientists or the doctors from the recombinant DNA. So that is the product that they will produce when they are experimenting the recombinant DNA. So let's move on the discovery of recombinant DNA technology. Discovery of DNA structure by Wilson and Crick in 1953. So last 1953, they discovered the DNA structure. Then isolation of DNA ligase in 1967. Then isolation of release in 1970. And then Paul Burge generated recombinant DNA technology in 1972. So during that time, Paul Burge discovered the recombinant DNA technology. So, kung ito 1972 dito, don't forget 1972. And then Cohen and Boyer 1973, they produced first plasmid vector that capable of being replicated within a bacterial host. So, this is the discovery of recombinant DNA technology. So, always remember that the recombinant DNA technology tells us how does it works in terms of their genes from different species through the process of the technology. Because the goal of the DNA technology, it is to reproduce a gene. So that carries genetic combinations and formations of value. So that was my report, my topic, and hope you will understand. And so now let's welcome my co-reporter, Mr. Warlito Yagas, for his report. So,
Now let's go to the steps of genetic recombination technology. There are 5 steps of genetic recombination technology. First is the isolation of genetic material. To isolate the desired DNA in its pure, free from other macromolecules, which means since the DNA exists in the cell membrane together with the other macromolecule such as RNA, lipids, protein, and etc., DNA must be separate from them. Then let's go to the second step is the restriction enzyme digestion. Restriction enzyme act as a molecular sensors that cut DNA at specific locations. In these steps, it restrict the incoming macromolecules to enter the purified DNA so that the pure DNA would not be mixed with other macromolecules from the outside. Then next is the application using PCR. PCR means polymerase chain reaction. It is a method of making multiple copies of a DNA sequence using enzyme. Since the restriction enzyme digestion is the steps where the purified DNA was incubated, and in this step it is where the pure DNA was copied and multiplied of thousands to millions of DNA molecules. For the continuation of my report, let me welcome my co-partner, Miss Roxanne Tyros.
thank you.
Thank you.
You go.
So I am the last, but not the least, reporter. So let me continue the previous steps. So here is the fourth and the fifth. I am the one who will discuss this. So in the fourth step, we have the ligation of DNA molecules. So the purified DNA and the vector of interest are cut with the same restriction enzymes. So they are being cut. The two molecules are being cut. So that, or the two fragments of DNA, and it will be ligated through the vector. So the vector will be the one who will. So the vector will be the one who will join the two fragments, the two DNA, so that they will be going to be to have a one product. So, and the process of joining in these two pieces together using the enzyme DNA ligase is ligation. So as what I have said, ligation, we are going to ligate the two DNA molecules so that it will become one. So the process of that in joining together the two fragments. So the process it is called ligation. So the last one, but not the least again, so the insertion of recombinant DNA into host cell. So this is to introduce into recipient host cell, most likely happen in the bacterial cell. So which means the two DNA molecules, the two fragment DNA molecules, as they are going to combine, or as they are going to connected, if they were going to join, and this time after that it will be transfer, or it will they will go to the host cell, or what I have said a while ago, the bacterial cell, and then that process will that process is called transformation, the transferring of that kind of product, or a kind of the DNA molecule that form. And remember that the that product, or I mean that DNA molecule will be ligated with the vector. So the vector here has a big important role in this process and the steps that we have tackled in this morning. So now let's proceed to the application of recombinant DNA technology. So these are the following applications where we can use the recombinant DNA, I mean. So recombinant DNA is widely used in biotechnology, medicine, and research. As we have observed, there are a lot of kind of vaccines that we are engaging nowadays, as we observe also that there's a viruses attacking in our places. So medicine, in this topic, I mean, the recombinant DNA technology is very useful, very applicable to the medicine. Yeah. So in the research also, there are a lot of research. There are a lot of researchers that using this kind of topic for finding some, or for making a experiment for the recombination of DNA. So another is recombinant DNA is used to identify, map, and sequence genes and to determine their function. So this recombinant DNA is very useful also, or applicable, in order to determine the sequence of genes, just like if what is the gene of your mother and your father, and so on to your onto the offspring, until to the last generation.
Next generation.
Yeah, to the next generation, I mean. So, and also this will determine the function of the gene. Next is recombinant proteins are widely used as reagents in laboratory experiments and to generate antibody proofs for examining protein synthesis within cells and organisms. So in this application, there are a lot of what do we call this? There are a lot of recombination of proteins which can help, just like for example, just like the beauty products, which can help us to have a
To glow.
for the anti-aging, just
Yes.
like that. There's a lot of combination for that, so that we can come up for a good quality product.
Dr. Bello.
Yes, Dr. Bello, that's right. So here are this is very applicable in the scientists or the researchers have a experiment for that. So this will help. This recombinant DNA technology is very helpful for, so that the organisms are so that we can, or they can examine the protein synthesis within the cells and organisms. So next is, let's proceed also to the limitations of recombinant technology. Actually, there are a lot of applications for the recombinant DNA technology, but we are just getting few of them, the important one. Then now let's proceed to the limitation of recombinant technology. So destruction of native species in the environment, the genetically modified species are introduced in. For example, human vaccination. There are people are being vaccinated, and there are people also are none. So it happened that it must be it must be separated, the vaccinated and the non-vaccinated. So if it is the recombinant technology, the recombinant technology is the one is it improvise. It means this is the improvised recombination of DNA. So meaning in the field of that vaccination or the vaccine, it must be or it will distract to the non-vaccinated people because the adaptations of that, or we do not know how they are going to fight the cells within the within cells of a person, or what are their what do we call that, the immune system, or they cannot fight for the virus. So it will distract. So it should be limited for the recombinant technology for some causes also. Okay, so next is the recombinant organisms or population clones vulnerable in exact same ways, just like what I have said earlier, that if the recombinant technology will be arise, there is a tendency that the people are also the same. They have an exact ways. They have the same what do we call in terms of, for example, in terms of immune system. If you are
Weak.
you are weak. So if you're being vaccinated, I don't know if what will be happen.
Possible
But
happen.
then there's a possibility that if there's an attacking of viruses, the non-vaccinated people will be affected. They are prone, or they are quickly to be affected with the viruses, yeah. And then
Creation.
another also is the creation of superbug is hypothesized. So the limitations of the recombinant DNA technology is that it should be when the scientists or the researchers will have an experiment to have a something experiment, it must be limited, or it must be what do we call that, hypothesized, meaning it will be more
Predict na.
it will be predicted, and it will be dapat
Possible happen. So that
they will know the
possible
possibility of what will be happening of that experiment if it will be going to arise, right?
Good ba na or bad?
It or whether it is
The
good
result.
or bad result. So next is the
Search.
such a system might lead to people having their genetic information stolen and used without permission. Yes, it is very what do we call that? Very
Common.
happen. So there are people that wanted to have that kind of gene because maybe they wanted to have, for example, the color of the skin. Some of the people wanted to have, if they are a morena or something, what do we call
They
that?
want
Brown color, not necessarily brown, but they wanted to become white, and they are going to get the gene of that people that have a cells which make them white. Yeah, I guess. Then
For example,
for example, the American, they're going to get some gene from the American and inject to the Filipina, or else just I guess
Black American baka.
yeah, and they will inject it so that they can they can provide a new gene, and it happened that there is no permission, and it will be stolen by the other people, and it's so very hurtful to us when we know that we have got something in our body and to transfer to another, and it's so very crucial to, yeah, that's maybe it will lead to the people genetic information scattered. Yeah, there is no another originality for that. Yeah, I guess, yeah. So next is last but not the least, many people worry about the safety of modifying food and medicines using recombinant DNA technology. So to sum up with, the recombinant DNA technology is very important, for this will help us how to know how to recombine anything that can make a new quality of something, or make something new, and through the help of the technology, through the help of the DNA, it will lead us also to have another
Journey.
exploration in our daily lives. And that's all for today. I hope you understand our discussion. So thank you so much for watching. Thank you for listening. And God bless you all.